Hello everyone. Good night. Welcome, first of all, to this first conference cycle in biotechnology. This is the last event, sorry, but I'm sure this is the icing on the cake, at least for me, a neuroscience person. So I'm super excited to be here tonight with you. I think it will be a great night, a great way to finish the day. So let me introduce myself. I'm Patricia Oliveira Silva. I'm the head of the Human Neurobehavior Lab at Católica, Porto. But today I'm here with another role. I will be your host for this third and final event of this edition. For sure, we will have more edition of this conference cycle. This is just the first one. But let me tell you a little bit about our guest today. Actually, it's a name I have talked about in my classes many times. So it's a huge pleasure to be here, to be your host for this specific conference. His name, as you know, is Joe Paton, and I'm sure you have heard about him, especially now that we have talked so much about time and the change in our perception in terms of time processing, the concerns we have in terms of some biases processing time. And he is at least now in Portugal, one of the most important reference in this area. So I start saying thank you so much for joining us tonight. It's a pleasure for Católica to have you here with us. He got his PhD in neurobiology and behavior at the Columbia University. Right now, he is the director of neuroscience research at the Champalimaud Foundation and the group leader of the learning laboratory at Champalimaud as well. In his laboratory, his main research work focuses on the neuromechanisms of learning, decision-making, action selection, and timing. And that's why his talk today is time is what your brain makes of it. So could not be more exciting for sure for all of us. Before sharing the stage with him or giving him all the stage, let me tell you that we would love to have you here with us participating in some way. So please don't be shy. Let us know what is going on. How are you using your time during this next 60 minutes? You can talk with us at the end, but don't wait until there. Please drop your questions here. As you can see, you have full access to the chat. So you can share with us. There is no wrong questions. The idea is really to open a conversation. So I'm sure our guests would love to listen a little bit about your feedback, comments, questions. That's why we are here. So I hope you can participate with us. But let's use our time in a very good way. So Professor Joe, please go ahead. It's a pleasure to have you here. Thank you so much.
Obrigado, Patrícia, pelo convite. A professora Patrícia falou que talvez alguns alunos estavam um pouquinho envergonhados sobre a possibilidade de falar em inglês. Quer dizer, eu falo português, eu vou dar a palestra em inglês, porque eu não estou acostumado a apresentar material científico em português, mas eu entendo completamente português, então fica à vontade, podes fazer perguntas em inglês ou em português, tanto faz. E eu sou muito informal. Se alguém não entende alguma coisa durante a apresentação, pode também escreve no chat para eu clarificar alguma coisa. Eu vou tentar ir devagar, porque não sei sobre o conhecimento de todos vocês, se vocês conhecem todas as técnicas e os termos e coisas assim. Então, fique à vontade de me interromper e fazer perguntas, por favor. Ok. Ok. Agora eu vou mudar para inglês. Okay, so as Patricia said, that the title of my talk provisionally was "Time is what your brain makes of it," and I will spend some time talking about temporal processing in the brain. But I'm actually going to switch it up because and hold on. And talk about a more general problem, which really sits at the root of my interest in how the brain processes temporal information. And that has to do with thinking about how the brain produces adaptive behavior. And my starting point for thinking about that problem is to consider that at the end of the day, what the brain is really for, what it evolved for, is for control, okay? It's for controlling the behavior and physiology of organisms in complex and dynamic environments. And temporal information is really important for that. And I'll come to that throughout the talk. But over 600 million years of evolution, nervous systems have evolved a number of different mechanisms for producing that control. So for example, if we look at a mammalian nervous system, like this cutaway of a rat nervous system, there are circuits in the brainstem, in the spinal cord, that are capable of relatively automatized forms of control. So things like reflexes, central pattern generators, movement primitives. And these mechanisms can be selected amongst, sequenced, or otherwise modulated by descending input from a number of different more adaptive control systems that are positioned more anterior in the nervous system. And I'm just highlighting three of them here in these different colors. The blue is the cerebral cortex. The purple is a collection of nuclei called the basal ganglia, which I'm going to spend most of the time talking about today. And then the green is a structure called the cerebellum, which contains half of all the neurons in your brain. And so, as I said, I'm going to focus on one of these brain systems, which is highlighted here in purple. That's the basal ganglia. Okay, but it's important to keep in mind that even though in the service of reductionism and making our problems tractable, we often focus on individual circuits, like I'll do today, these circuits function in the context of a larger system where there are multiple brain systems. They have to interact in order to ultimately support the behavior of an organism. Okay, and I'm going to talk a little bit about how our studies that started with an interest in temporal processing have begun to make us think about the kind of multiplicity of control that the brain brings to bear on the problem of adaptive and intelligent behavior. Okay, but I'm going to start off by giving you kind of an overview of the general approach we use. As you can already probably guess, we use animal model organisms, okay? And it's useful to think about kind of what paths information can take through the nervous system, okay, in these organisms. So if we zoom in here, okay, you can imagine that information can take a relatively shallow path through the nervous system. For instance, reflexes, right? When you go to a doctor's office and the doctor hits your knee and you reflexively, jerk your leg, that's a very short circuit that goes from muscle stretch receptors into your spinal cord and then back out to motor neurons almost immediately, okay? So it's a very short path. You can take an intermediate path. So imagine you have information coming in through a sensory system. It comes up through the spinal cord, goes to the thalamus, might go up to a primary sensory cortical area, okay? And then it might jump over pretty quickly to a motor area and then get fed down either back to the spinal cord and out to motor neurons in the muscles, or it might go through one of these so-called subcortical structures, okay? And then make its way out to the motor periphery. Okay, so that'd be like a path of intermediate depth, of intermediate length. Information can also take really, really long, tortuous paths through the nervous system. So for instance, you might come in to the spinal cord, you might go into the thalamus, you might go to a low-level sensory processing cortical area, and then information might be passed through a series of hierarchically organized brain areas that basically build representations of the outside world of increasing complexity, abstraction, temporal extendedness, okay? From there, that very high level information, for instance, the identity of the person who's sitting in front of you, that's a very abstract kind of representation, might be sent over to some high level cognitive area in the front of the brain. Okay. And from there, it might come down into one of these subcortical structures. From there, information can actually take these really torturous kind of spiral loops from these subcortical areas back into this cortical area, back down, back up again, before finally, after a lot of processing, that information making its way back out to the motor periphery. Okay. Now, why am I pointing out all these different kinds of paths that information can take? Well, one of the main points I want to make is that the brain is complicated. Okay. There are order 100 billion neurons in your brain. Okay. Equal numbers of glial cells that sort of support the functioning of those neurons. So understanding a system of that kind of complexity, okay, with this many different paths that information can take, requires more than just observing it. Okay. You need some sort of theoretical framework. Okay. And so one central kind of component of the type of research we do in my lab is that our experiments are guided by hypotheses that come from computational and theoretical frameworks for thinking about behavior. Okay. And you can see why such frameworks are important from this quote by Thomas Young. So I don't know how many of you know who Thomas Young is. He's sometimes described as the last man who knew everything. He was a physician in England, the end of the 19th century. He was also a physicist, a Egyptologist. He's responsible for proving the wave theory of light. Anyway, he says, although the invention of plausible hypotheses, independent of any connection with experimental observations, can be of very little use in the promotion of natural knowledge, yet the discovery of simple and uniform principles by which a great number of apparently heterogeneous phenomena are reduced to coherent and universal laws must ever be allowed to be of considerable importance towards the improvement of human intellect. So this idea of using sort of mathematical frameworks, theory, to come up with a set of simple and uniform principles that sort of make compact the description of a very complex system. That's at the heart of the approach that my lab uses. And the framework we use is that of reinforcement learning, which I'll get into in a little bit. Okay, so as I mentioned, I'm interested in adaptive behavior, and in particular, we focused a lot on understanding how it is that dynamic internal representations are generated by neural circuits and then translated into behavior. And that's because it's these kinds of internal dynamic representations that underlie our ability to think and to plan. They're generated when we apply rules and so on and so forth. But the fact that they're internal and they're dynamic, they're changing, is both what makes them interesting, but also hard to study. So to make these kinds of questions more tractable, we train rodent model organisms to perform carefully designed behavioral tasks that require these kinds of dynamic internal representations and bring them under experimental control. And then while they're performing these behaviors, we go in and we measure the activity of electrically or otherwise genetically or anatomically targeted populations of neurons. This is an example of one such measurement. These on the top here is, let's see, can you see my pointer?
Yes, we can.
Okay.
Yeah.
So on the top here is called a raster plot. Each row here is a so-called trial in a behavioral task. And each little black tick mark here, okay, represents a single neuron that's emitted these all or none electrical impulses called action potentials, okay? And so down below here, you can see a histogram of those action potentials, okay? Aligned on, in this case, the application of a light stimulus in the presence of an opsin. So it's a protein that's light sensitive that ends up inhibiting the neuron in the presence of light. So here we're just showing that you can inhibit a particular genetically targeted neuron during behavior. So we either measure, we manipulate these genetically or anatomically targeted populations of neurons during behavior to try and understand, how are those neurons involved in producing that behavior. And of course, as I mentioned, we use theory as a guide. And so we often construct models, computational models of circuit function and of behavior, and then relate what we see in the behavior and what we see in neural activity to the components of those models in this sort of iterative interaction between mathematical frameworks and experiments. Before I get into the specifics of the experiments, I want to thank the people who actually do the work. Okay, I'm going to touch on a number of people's work today. Actually, not everyone who has an arrow, but most of them. I want to also thank the excellent scientific and technological platforms we have at the Champalimau Foundation, without whom our work wouldn't be possible, our funders, and also our collaborators. Okay, so how does the brain produce behavior? Well, that's a complicated question, but in the context of reinforcement learning, decades of work has kind of led to the development of a sort of dominant view of how particular brain systems interact, such that animals can learn through interaction with the environment how to behave. Okay, and I'm going to give you just a kind of 30,000-foot view of what that dominant view is with respect to the interaction between a couple of these brain systems. Okay, so here's a schematic of a sagittal slice of a mouse's brain, and I've just highlighted in red here this collection of brain structures that collectively are called the basal ganglia. Okay, and the dominant view that's evolved about what the function of the basal ganglia is for behavior goes as follows. So the idea is that inputs from a really wide range of different brain structures in this overlying area called the cortex, as well as the thalamus, which is this gray area in the center, provide the basal ganglia with information about the sort of state of the world that an animal encounters, the situation the animal's in, as well as the actions that are available in that situation. And this state and action-related information converges in the major input area of this brain system called the basal ganglia, which is called the striatum, with extremely dense input from a collection of neurons that are in the midbrain that use the neurotransmitter dopamine, which many of you may have heard about being associated with things like reward and addiction. Okay, and these neurons are known to carry an important teaching signal within computational models of reinforcement learning called a reward prediction error. Okay, and it's the convergence of this teaching signal with this state and action related information that's thought to produce plasticity, so changes in the strengths of connections between neurons in the striatum, that allows it to learn something about the value of available actions in a given context. And just to give you an intuition about why a so-called prediction error is a good teaching signal within these models, if you think about it, if you're surprised by something, okay, so there's an error in your prediction, that surprise is a good indication that there's something more to learn. If the next time you come into that situation, you want your predictions to be more accurate, okay? So that's the basic intuition behind this now very famous teaching signal carried by these dopamine neurons, and why people think that they update learning about value, because they encode errors about the prediction of reward. So if you reduce your errors in predicting reward, okay, you can imagine that that can be used in the context of control to basically choose actions that are going to lead to the most reward. Okay, and that's the whole idea behind reinforcement learning. Okay, it's that it gives you a set of mathematical tools by which artificial agents, or in this case animals, can learn through interaction with the environment what actions they should take in order to maximize reward. All right. And the idea is that the basal ganglia can then influence the selection of actions through a number of different extrinsic connections that it has with downstream motor areas, either in the brainstem or indirectly back in this overlying cortical area through the thalamus. Okay. Now, the basal ganglia is also associated with a number of different disorders. Okay, so in the context of movement disorders, you have diseases like Parkinson's disease or Huntington's disease. Okay, Parkinson's largely results from the loss of a certain subset of these dopamine neurons that project to the striatum. Okay, Huntington's disease, at least early on in the disease progression, is characterized by the selective loss of a cell type, which I'll tell you a little bit about today, in the striatum. Okay, so there's this notion that the basal ganglia is involved in selecting actions, is sort of broadly aligned with what we see in terms of movement disorders of action production, when you have dysfunction in different elements of this circuit. But it's not just limited to movement, because basal ganglia dysfunction is also associated with other different kinds of disorders, such as impulse control disorders, so attention deficit, hyperactivity disorder, addiction, OCD, or Tourette's. These would all be considered disorders with a significant impulse control component. Okay, and that'll become that's very related to some of the results I'll tell you about next. Okay, but you can kind of get a sense of how this framework of reinforcement learning, it's coming out of sort of machine learning, and it's actually behind a lot of recent advances in artificial intelligence today, is not inconsistent with the symptoms that we see when there's dysfunction in this set of circuits. Now, another interesting and prominent feature of the circuitry of this brain system is that, similar to how I described that general example of how information comes in to the nervous system and then can take various different paths through the nervous system before going out to the motor system. The basal ganglia is organized in a parallel circuit architecture. So this is now a schematic from the human brain, okay, but this applies across all mammals. Inputs from, say, low-level sensory or premotor areas, they'll be passed through, one set of circuits, whereas information from more cognitive areas in the more frontal cortical areas, they'll go through another set of circuits, and those from more limbic areas that are associated with, say, emotional processing, they'll go through yet another sort of parallel circuit path. And this information is fed through in a more or less segregated manner. Okay, and yet within each of these circuits, there's a very similar kind of microcircuit architecture, which suggests that, there's certain things that are wired up to perform particular computations. And generally speaking, they're consistent with that overarching model that I described of reinforcement learning. Okay, you have a lot of dopamine input. Dopamine can produce synaptic plasticity. It seems to aid in the selection amongst information that you have coming into the system, but it's operating on very, very different kinds of information. Okay. So here you can see a reflection of that comes now from the mouse. This is just a schematic from a recent high throughput circuit tracing study. So now we have all sorts of tools for being able to trace neural circuits through multiple synapses. Okay, and the conclusion of this paper was that in the mouse, you have essentially at the output 6 separate different channels, which integrate information from very different sets of outlying cortical areas. Okay. So this parallel circuit architecture is a really prominent feature, and it figures prominently in some of the work I'm going to tell you about. Another prominent circuit feature that doesn't show up in these computational models is the fact that there are 2 main feed-forward circuits in this brain system, okay? The so-called direct and indirect pathway, right? So in this major input area of the basal ganglia called the striatum, roughly 90 to 95% of the neurons are so-called projection neurons. So they send connections outside of the striatum. And amongst those 90 to 95% of neurons, you can further subdivide relatively evenly these neurons into two classes, okay? The direct pathway neurons are so named because they send information directly to the output structures of the basal ganglia, okay? And because these neurons are inhibitory, okay, they use a neurotransmitter called GABA, which is inhibitory. It decreases the activity of neurons downstream. When you have activity in these neurons, that would inhibit neurons that are sending outputs from the basal ganglia, thus disinhibiting basal ganglia targets, okay? The indirect pathway, because of an extra set of synaptic stages, has an opposite effect on the output of the basal ganglia, okay? So there are these two main feed-forward circuits. They seem to have this sort of push-pull influence on the output of the basal ganglia. And, you know, for a long time, people really didn't understand how this dominant reinforcement learning inspired view of the basal ganglia, how does that interact with the presence of these kind of push-pull circuits, okay? So in the first part of the data that I'm going to present to you, I'm going to talk about a project that sort of pulls together these two features of the circuitry of the basal ganglia, parallel circuit architecture, and this two main feed-forward push-pull kind of circuitry, and starts to build the picture of why or what are the consequences of those features of the organization of this system. Now, I said these two pathways look like they have a push-pull relationship, but when people started to be able to record the activity of neurons in these two pathways, they were somewhat surprised to see that when you look around movement, both pathways are active, okay? So here's an example from a study about 10 years ago, where activity was recorded from neurons in the direct pathway or indirect pathway aligned on a particular movement. So this is in mice, they're pressing a lever. So this is aligned on when the mouse presses a lever. And you can see that in both pathways, okay, there's a peak in activity. So some people looked at this and they said, wait, this is confusing. It looks like they should have opposite effects on, say, movement, okay, but they're both active when the animal is moving. So what's going on? Well, it turns out that for decades now, people have been looking just at the anatomy of the system and sort of hypothesizing that perhaps producing a movement, okay, controlling your behavior, involves not just selecting particular movements, but also suppressing other ones, right? And if you step back and think about it, a lot of our behavior is actually shaped by our active suppression of some behaviors and the kind of elevation of other ones, right? So maybe movement involves both promoting some action representations or motor programs and suppressing other ones, right? Well, if that's the case, we reasoned you shouldn't look when animals are moving for signs of this opposite function, but you should maybe train an animal to actively suppress the urge to move. And there, our prediction is you should see this real difference in the activity in the two pathways. Okay, so to test that idea, we adapted a behavioral task that we've been using for a long time to ask a number of different questions, some related to temporal processing, and I'll get to that at the end. But in this case, we're really using it to ask questions about motor control. And the way we adapted the task is that we force the animal to stay still, to suppress the urge to move, okay? But the basic version of the task goes as follows. Mice are placed in a box where there are 3 nose ports positioned along a wall, okay? And these nose ports have sensors in them. We can deliver rewards through them, okay? And they learn to initiate trials by inserting their snout into a central port. That elicits a brief auditory tone, a brief sound, followed by a variable time delay before a second tone is played, okay? And at this point, animals are free to move to the left port or the right port to get a reward, just a liquid reward, depending on whether they believe the time delay between those 2 tones was shorter or longer than a learned category boundary, okay? So basically, They learn that if the intervals are shorter than 1.5 seconds, they have to go to the left to get reward. And if the intervals between the two tones are longer than 1.5 seconds, they have to go to the right side to get reward. Okay, so that means that they're making kind of categorical judgments about whether intervals are longer or shorter than 1.5 seconds. Now, going back to the original point I made about studying kind of internal dynamic representations, here we can see how this kind of task, which is ostensibly focused on looking at time processing, okay, is actually, it's an exemplar of a task that requires animals to make use of a dynamic internal representation, okay? Because at the time of the second tone, there's nothing in the environment that's telling the animal where to go to get reward, okay? Instead, they have to rely on this kind of internal evolving representation related to elapsed time if they want to maximize the amount of rewards they're going to get. And they can learn to perform this task quite well. So here's a representation of their behavior. On the y-axis is the probability that the animal makes a long choice as a function of the different intervals we present to the animal, okay? And you can see for the so-called easiest stimuli, those that are furthest from the 1.5 second boundary, animals perform almost perfectly. So they're almost always choosing long for this 2.4 second stimulus. They're almost always choosing short for the 600 millisecond stimulus. But as the intervals approach this decision boundary, their choices become more variable. So sometimes they're judging the interval as longer and sometimes shorter than 1.5 seconds. And these kinds of psychophysical tasks have proven powerful for kind of probing the mechanisms of perception and cognition, because they give us things like a quantitative description of the sensitivity of animals' judgments to some dimension of the environment, in this case, time, okay? But more generally, they give you this scenario where from trial to trial, animals are making different judgments about the same condition, okay? And that variability in the animals' judgments and their decisions is a lever we can use to then go in and interrogate the nervous system, because we can ask, where do we see neural signals that correlate really tightly with the animal's judgments, and where, when we perturb the function of neural circuits, do we see changes in the animal's decisions. By the way, I can't see the chat, so if anyone has a question, just speak up. Okay, so the way we modified this task to ask the question about action suppression was we required that the animals stay in this center port during the entire interval. Okay, so that's up to 2.5 seconds roughly. And that's actually pretty hard for mice to do. They're relatively impulsive, okay? And if they left the center port prematurely, then they would get a white noise auditory stimulus, the trial would be aborted, and they wouldn't have the opportunity to get the reward, okay? So to maximize reward, they really needed to kind of force themselves to stay still in the center port. And indeed, they could do that. So here you're seeing a representation of their speed. We're just tracking the animal's position in the box using a high-speed camera. You can see there's a peak in their movement velocity as they move to the center port to initiate the trial. And then after a sort of postural adjustment, they enter this period of relative immobility during the interval presentation. And then when the second tone is played, there's another peak in velocity as they move to execute their choice. Okay, and we didn't think they were sitting in this period here passively. We thought they might be kind of actively planning initially a movement to the short side, and then once they believed that time had elapsed past the 1.5 seconds, they would plan a movement to the long side. Okay, and we actually saw evidence in their behavior that that's what they were doing. So I mentioned it was difficult for the mice to actually suppress the urge to make a movement for up to 2.5 seconds. And in fact, roughly 30 to 40% of the time, they failed to suppress the urge to make a movement. And when they failed to suppress the urge, they left the center port prematurely. And most of the time, they end up making relatively normal choices. Okay, so they look outwardly like any other choice the animal might make, right? And they have structure. So here's the probability that the animal so-called broke fixation, which means they failed to suppress the urge to move as a function of time in this delay period. conditioned on whether they subsequently went and made a short judgment or a long judgment. You can see that when they broke out early, when they failed to suppress action early, they most often made short choices. And when they failed to suppress action late, they most often made long choices. Okay? So we take this as evidence that what's going on is that in the animal's brain, there's a sort of latent, hidden internal movement plan, okay, to go to one side early, the other side late, and they have to actively suppress that plan until they reach the appropriate point in the trial, at which point they can make their choice. If they fail to suppress it, then they let fly whatever action they had been planning. So now that we have them in this state where they're actively suppressing the urge to make movements in one direction early, another direction late, we can ask our question about what's going on in the direct and indirect pathway. So here I'm going to show you some neural activity data. So this is data recorded using a calcium imaging technique. So there's a genetically encoded calcium indicator that fluoresces in the presence of calcium and when it's excited with the right wavelength, okay? And we're just collecting the fluorescence through a fiber optic that's placed into the sensory motor region of the striatum, okay, where these direct and indirect pathway neurons are. So it's sort of like a cell type specific, so direct or indirect pathway neurons, pooled measure of activity of neurons in each of these pathways, okay? So aligned on the initiation of the trial, when the animal was moving to initiate the port, you can see, just like I showed you in those previous publications, there's a peak in activity in both pathways. But then when you get into this delay period where the animal has to suppress the urge to move, you see relatively privileged activation of the indirect pathway, whereas the direct pathway hits the floor. So this is consistent with our hypothesis, right? We said, well, maybe you're going to see distinct patterns of activity in direct and indirect pathway if you actually train the animal to suppress the urge to act, because now you should be pushing the system towards generally suppressing movement. Excuse me. Whereas when the animal's moving, it's going to be a combination of suppressing and promoting. Excuse me. I've had sick kids this week, so I think they may have gotten me. Okay. So this is pooled over both hemispheres. But we wondered whether we could see some evidence of sort of the suppression of specific actions by looking at one hemisphere at a time. And so I'm going to refer to the hemispheres in relation to the categorical choice. So either the hemisphere that's contralateral to a long choice and contralateral to a short choice. And for those of you who haven't had a lot of neuroanatomy yet, the reason is that generally speaking, above a certain level of the brain, it's organized such that one hemisphere is more concerned with either stimuli or actions to the opposite side of the body. Things get crossed, okay? So you can think about contra long and contra short as neurons that care about the long movement or neurons that care about the short movement, okay? So what do we see when we look at one hemisphere at a time? Well, if we look at the hemisphere that's contralateral to a long movement, we see in the indirect pathway, the suppressive pathway, activity is steadily climbing during the delay period. If we look at the direct pathway, activity is dropping. If we look at the other hemisphere, we still see the overall dominance of the suppressive indirect pathway, but now, you know, it's the modulation is lesser and it's sort of dropping over time. And we looked at this and we said, huh, that's interesting. There's this asymmetry between the two hemispheres. Now, why might that be? Well, if you think about this task, there's an asymmetry in it, right? Imagine you're the mouse and you're doing this task and time is going on. And at the beginning of the trial, you don't know whether you're going to have to go to the left side or the right side to get reward. But the more time passes, the more you become certain about where you're going to have to go. Specifically, when you know time has gone past 1.5 seconds, you know exactly where you're going to have to go to get reward. And your job becomes, okay, I just have to hold my horses until I get the go cue, and then I can go and make that movement. And so we hypothesize the animal might be actually more tempted, okay, to make a movement late in the delay period towards the long choice than they were to make a movement to the short choice early. And maybe that's why you see this asymmetry in modulation, because the brain actually requires more kind of suppressive control on to suppress the urge to make long choices later than it does to make short choices early. And you can see that in their behavior as well. So if you look at the probability that the animal breaks fixation and makes a long choice, okay, as a function of time, given that they haven't broken fixation yet, you can see it skyrockets after the decision boundary. Whereas you look at that same characterization of behavior conditioned on making a short choice, and the modulation is much more modest, okay? And these kind of match, right? So there's a sort of nice match in the degree of modulation in the activity of these two brain circuits, and this behavioral indication of the urge or temptation that the animal has to make a movement. I'm going to kind of speed up here because I'm running out of time. But we saw that activity was disrupted, particularly in the indirect pathway when the animals failed to suppress movement, okay? And generally, we saw this sort of pattern of opposite lateralized dynamics across the two pathways. So people hadn't really shown that you can get opposite patterns of modulation and activity in these two pathways. And we were able to show that by looking not when animals are moving, but when they are actively suppressing the urge or the temptation to make a movement. We then went and, or when I say we, I should say Bruno Cruz, who is a very talented graduate student who's since graduated, who did all these experiments. And he used a technique called optogenetics, okay, which involves putting one of these light-sensitive channels into neurons through using viral vectors or sometimes genetic techniques. And that means you can just shine a light through a fiber optic into the brain, and you can manipulate the activity of specific genetically targeted populations of neurons. So Bruno did this here, targeting indirect pathway neurons and inhibiting on one side of the brain at the time. And here he's just plotting the probability that animals made failed to suppress movement as a function of time. On trials where he inhibited these indirect pathway neurons, in comparison to trials where he didn't, in black. And you can see that all the excess broken fixations, so the animals were more likely to fail to suppress movement, and they were particularly more likely to fail to suppress movement at the times when that hemisphere was dominant, okay? Even though the light was on, inhibition was on during the whole trial. If you did the same experiment on the other hemisphere, now you see all those excess failures to suppress movement, they're happening late, okay? Again, at the time when this hemisphere, that contains the long neurons, was dominant, okay? So this is really compelling evidence that the indirect pathway of the basal ganglia is sort of dynamically recruited. Neurons on one side of the brain are recruited early on to suppress the urge to move to the short port. And then as the animal's sort of internal timekeeping tells it that time has passed the decision boundary, then another population on the other hemisphere gets recruited to suppress the urge to make a movement to the long choice, okay? So when you go back to that slide I showed you at the beginning, and I showed you a list of sort of impulse control disorders, you can start to see how these kinds of experiments in mice might give us insight into the circuit mechanism by which the basal ganglia is contributing to good impulse control, okay? Okay, so I think I'm going to skip over this. but I'll just give you a kind of intuition. So I mentioned we build computational models. Okay, so Gonzalo Guiomard, who is a graduate student in the lab who's now finishing up his PhD, what he did is he built a computational reinforcement learning model that contained elements of the circuitry that we know of in the basal ganglia. It had so-called synaptic plasticity rules that were drawn from experimental data. So there's different plasticity onto the direct and indirect pathway. Dopamine influences those two pathways differently. Okay, and what the upshot of what he found with his model is that he couldn't explain the broad set of data we were seeing without assuming that essentially the basal ganglia was acting almost like a so-called multi-agent system. Okay, so these parallel circuits that I mentioned, they seem to be learning different kind of programs for behavioral control that interacted with each other. It was almost like, you know, if you had a collection of people in a room and they were all trying to play a game, each of them might learn different strategies, but those strategies would depend on the strategies of each other. Okay, and so what Gonzalo showed is that he could reproduce broadly the pattern of failure to control impulses. He could produce a model that could perform the task. He could reproduce the patterns of neural activity we saw in the experiments, and he could reproduce the optogenetic inhibition experiments by essentially going in and manipulating the activity of specific elements of this model. Okay, and the thing we're really excited about this model is that it brings together, again, you know, these two really prominent features of basal ganglia circuitry, that is the parallel circuit architecture and the presence of this push-pull kind of circuitry, and gives you a sense of the consequences of those different features of the organization. Specifically, we see that one parallel pathway learns to suppress the urge to move that another parallel pathway develops through its own learning process. Okay, so in the last few minutes, I'm going to sort of change gears a little bit and go from questions related to motor control to those that are related to temporal processing. So is there a question?
No, that was just a glitch.
Okay. Okay, so in order to, what just happened? So in order to perform this function of kind of learning which actions predict the most reward, the algorithms that we think that the basal ganglia are implementing, they require information about time. Specifically, they need to know what has happened and when it's happened, okay, in order to build up these kinds of predictive representations that underlie these programs for behavioral control that come out of reinforcement learning models. Okay, and so so I'm showing here just a representation of how those show up in the computer models. Okay, this is a representation of time where you have these different functions that essentially represent that something has happened at different delays into the past. Okay, and we know that brains can't do this. Okay, your brain doesn't have a perfect clock, right? The fact that clocks exist already tells you that your brain can't do it, right? Because if it could, then you wouldn't need those clocks. So one of the first questions we were interested in when I set up my lab is, well, how does the brain get access to information about what happened when? Okay, and I've already told you about this task we used to ask that question. The first thing we did is ask whether the striatum was necessary for performing this task by pharmacologically inhibiting the function of this area, and we saw a marked decrease in animals' performance. I should point out that if you inhibit the basal ganglia, animals don't move. Okay, so this is at levels where animals are still doing similar numbers of trials. They look in terms of motor function to be normal, but you still see a decrement in performance. We also suspected that this major neuromodulatory input from dopamine neurons might be playing a role in temporal processing. Patricia at the beginning alluded to the fact that there are specific pathological states that are associated with deficits in temporal processing, and some of those affect the dopaminergic system. So we did a series of experiments, or I should say Sofia Suarez did a series of experiments where she showed that the activity of midbrain dopamine neurons, the variability from trial to trial, correlated with variability in animals' judgments. And if she manipulated the activity of those neurons, she could actually push animals' judgments around. So timing behavior is dependent on this area and one of its major modulatory inputs. And so what kinds of signals, in the striatum might underlie the necessity of the striatum for performing this time-dependent behavior. And this is just to give you an intuition of what we found, okay? So I'm going to show you just a representation of actual neural activity. Okay, it's an animation. And each little circle here is going to represent the activity of an individual neuron, okay? And you're going to see that their activity is variable over time, okay? So you can actually reorder these cells, okay? And if you do that, you can see that, activity is kind of moving in a bump around this population, right? And so we made this animation just to make it sort of clear how one could extract information about the progression of time by just sort of looking at where activity is in this population, okay? And that we've kind of confirmed in a number of different experiments now, and really, I think, represents a general principle of brain function. That is that the brain gets access to information about the passage of time through the kind of intrinsic dynamic patterns of activity that populations of neurons tend to produce. Okay. Now, many areas of the brain are going to do this. Is the animal actually using it to guide their behavior? One way to look at that is to ask whether that trial-to-trial variability in animals' choices correlates with changes in speed of this bump of moving activity. Okay, and you can kind of conceptualize this trajectory of neural activity in so-called neural space. So now imagine, instead of looking at all those neurons, you're looking at the activity of 3 neurons. So each of these axes would represent the firing rate of an individual neuron. So how many action potentials is it producing per unit time? And then during the interval presentation, activity might start here. And then as the activity of this neuron goes up, the state of neural activity is going to move, and it's going to follow a particular trajectory in this space that's defined by the activity of these 3 neurons. Now, of course, you can think about neural space as being much higher dimensional. Instead of 3 neurons, n neurons, however many are in the population you're recording from. And so what we did to ask whether the variability in the speed of population activity correlates with animals' judgments is we looked at the state of activity from a population of striatal neurons at the offset of an individual interval, say this interval here that's near 1.5 seconds, okay? And we'll schematize that as this point in this space here, okay? So this just means there's a certain set of activity across these, say, 3 neurons here. And then we can project this population state onto this typical trajectory that activity normally takes, right? And that gives us a number, okay? If you quantify what fraction of this trajectory has activity surpassed at the offset of this interval, that gives you a number that quantifies how fast population activity has evolved on that trial, okay? And then we can ask our question, does that speed metric, does it correlate with the animal's judgments? Okay, so from trial to trial, this will fall onto different positions, right? And we can ask, if we look at cases where, say, the speed was average, what's the animal's probability of making a long judgment when it goes faster or when it goes slower, okay? And when we did that, we saw that there was this systematic relationship between the speed with which patterns of activity evolved on individual trials and the animal's judgment. Okay. If you look at all the different intervals, you can see that this is systematic. Okay, so this effect is biggest for these intervals that are near the decision boundary, and it's negligible for the easy stimuli. So this is consistent with this kind of evolving pattern of activity forming an internal representation that the animal is actually using to guide its judgments. Okay, but this is just correlational, right?
Sorry. So sorry, but I would love to listen to our audience as well.
We have to
So
stop.
1 minute to close. Amazing.
Yeah, can you give me 1 more minute?
Yes, sure. Go ahead.
Okay. So, as you've all probably learned, correlation does not mean causation, right? So in order to really test whether animals are using this, you'd like to manipulate how fast or slow activity is evolving and ask what happens to animals' behavior. Okay, so I'm going to fly through this, but long story short, we made a device that allows us to manipulate focally the temperature of striatal tissue. And we saw that we could change the temporal scaling of neuronal responses, so it could stretch and contract neural activity. Okay, these are 2 example neurons where you can see we're contracting them with warmer temperatures, stretching them with cooler temperatures. Okay, this was very consistent. And if you look at the animal's behavior, we're able to stretch or contract their choices. It's very robust. It's in all the animals. Okay, so this is actual causal evidence of this principle that that the speed with which populations evolve provides a temporal basis for, in this case, a decision. But the last thing I'll leave you with is that, you know, I mentioned that there are different brain systems, okay, and they seem to have different stereotype circuit architectures, suggest they have different functions. And we think of the basal ganglia as a motor system. So we thought maybe we might see an effect on the speed of movement, okay? But when we changed temperature, but we didn't. Okay? We didn't see changes in their path. We didn't see changes in how fast they moved, okay? And that was in stark contrast to these really systematic changes we saw in their decision, okay? So this is just to say that time is really fundamental for many, many different things the brain does, okay? And we're doing experiments that are starting to point out what are the specific things that specific brain systems are contributing to behavioral control. In the case of the basal ganglia, it seems to be a kind of discrete kind of control. And we think that other brain systems, such as the cerebellum, are likely brought in for more continuous forms of control. Okay, so with that, just to recap what I told you. So I gave you an overview of this idea of neural reinforcement learning, theory-inspired experiments to try and understand how the brain produces adaptive behavior. Pointed out that there are some gaps between biology and algorithms, and this is sort of a core part of what my lab does. It tries to fill those gaps. And I told you a couple different stories related to both motor control as well as how the brain gains access to a temporal basis for behavior. So thank you.
Wow. Thank you so much. Thank you for being here. I'm so sorry we don't want to go over the scheduled time, and I know it's time to go out at least of this academic part, but I really want to say 1 or 2 words. First of all, I'm sure it was challenging for some of our students here, but sometimes our brains need challenging food. So I think it's fascinating to see basic research about that, to understand how we are able to talk about brain involvement in behavior, right? To do this type of translational neuroscience, we need to know this type of research and understand how can you manipulate animal models to do that. So I hope you appreciate that as I do. It reminds me when I'm talking with my students about the ions movement, through the neuron membrane, and they say, no way, we are not that simple. Oh, yes, we are. Actually, we are. Everything we are talking about ions, we are talking about those circuits, but that's the magical happening in our brains, right? I know it was difficult for some of you to follow the discussion here, but that's part of the journey. So it will be amazing, at least to open for one very quick question. So if we have here any candidates, we can take advantage of Professor Joe's presence. Someone?
Well, while we're waiting, I would just, there's a hand raised. But I would just say, the idea is, as Patricia is saying, there are really simple processes that you can understand if you focus in on them. But when you iterate them, hundreds of billions of times, right, you get these sort of emergent properties that we know of as, things like intelligence and consciousness and all of these, and emotion. So those two things are not inconsistent with each other. To have relatively simple, compact descriptions, principles for how things work, and to understand that they give rise to this really complex functions that the brain is able to produce. Yeah, sorry. So there's a...
I fully agree. Amazing. Raquel Claro, please.
Good evening. Thank you, Joe, for your presentation. It was very hard, just like teacher Patricia was saying, it's very hard. But I got especially interest about one topic that you talked about, which was decision making. I specifically enjoyed that topic, and I wondered when you talked about the different hemispheres and the output that you have, and you said that, for example, it's hard when the rat don't knows where he goes, and that maybe is an explanation why different outputs from different hemispheres. And I was thinking about, what about human life? Would this output, would you expect to have this output, for example, on human? Or is this output only applies to decisions that involve movement? For example, imagine if we are talking about behavioral, pro-social behavior on humans, what would you expect of humans in this case?
Yeah, so, we often are using like these motor assays in experiments because they're the easiest to characterize and the most accessible, right? But one of the most attractive things about this framework of reinforcement learning for selecting actions, when you're talking about selecting actions, you're talking about decision making. Do I do this thing or do I do this other thing, right? Is that it's very general, right? So depending on what actions are on offer, right, these same algorithms can be used to drive decisions about whether to move to the left or the right, or whether to eat that food or this other one, right? Or whether to engage socially or support someone, or be competitive. That's what we think this parallel circuit architecture subserves, is that you have different kinds of information being pushed through these same algorithms, the same basic learning process, okay? But it's applied to different kinds of information, right? And so you can even see this in the motor system, different kind of levels of abstraction, right? When you think about space, right? Like I can choose my actions, like go to the right or the left of my body, or I can choose my action in terms of a location in space, right? Like I want to go to the corner, right? And those are very different kinds of spaces. So that's a very concrete example of different kinds of action spaces. But then you can use that as a springboard for thinking about even more abstract action spaces, right? So thinking in general, right? Thinking is a series of selected internal actions. You can think about it that way, right? So as you're in a state in your mind, you're considering a couple different options. These same kind of reinforcement learning algorithms can be used to basically decide, well, what is the next state I want to go into? But it's internal, right? So yeah, it's a great question. And these same kinds of models, these same kind of mechanisms can be used to support a wide range of different kinds of decisions.
Wonderful. Thank you.
Yeah.
Thank you so much. I'm sure we would have many other wonderful questions as well, but I'm assuming you can share with us. You can use our emails, to share comments and questions. It's a pleasure for us to disseminate what we do. I'm sure we share the same feeling, right, about that. So I have no words to say thank you to you. It was fascinating, as I told you. I had a great time. I hope you too. We apologize for, crossing our barrier of 8 p.m., but it was for a very good reason. So thank you.
No, I'm happy. I'm sorry I went on, I tried to go slowly, so I went on a little bit longer than I had planned, so I didn't give enough time, but really it's no problem, and thanks a lot for the invitation. And if any of you are interested in if this was hard to catch, you can look at the talk later, but you can also Patricia, if you want, you can write me and I can send you some kind of reviews that will give a little bit more context if people are interested.
Wonderful. I will for sure. So thank you so much. Thank you all. So hope to see you soon in another initiative here in Católica. For our dear students, thank you so much for joining us today, and hope you can participate in the next sessions as well. Thank you so much. Bye-bye.
Thank you. Bye-bye.
Thank you. Take
Bye.
care.
Thank you.